Welcome you all in the class of Computational Biology and Bioinformatics. So students, on the last turn we have discussed about the Genome-Wide Association Studies. Means introduction of the Genome-Wide Association Studies. Okay. So today we will discuss about the details of the and steps of the Genome-Wide Association Studies. What steps it includes. So I am revising again. This is the introduction. What we do in the genome-wide studies, we analyze a lot of genomes of different populations. Okay, and that is useful for the analysis and diagnosis of the disease. So in the medical areas, it has importance, great importance. The genome-wide association studies. We have already discussed about this. So generally we analyze the genome that is associated with disease, because in the human we have lot of disease, genetic disorders. So study of the genomes is necessary. So we can analyze the genomes of different populations. Okay. So in the last classes we have also studied about the SNP variations, genetic variations that play an important role. But what we do in the genome-wide studies, so we do the studies at the large scale. That is the difference. So we apply lot of techniques in the combination. Okay, so genome-wide studies, association studies consist of different steps that we are going to discuss today. On the last turn also we have discussed about these things. So this is very difficult and frustrating sometimes when we have lot of things and you want to identify the candidate genes. Okay, so we need the comprehensive method to study that, to study the traits of the characters. So from here we are starting today. We are discussing some more details. So a genome-wide association studies test hundreds of thousands of genetic variants. Okay, across many genomes. So to find out the traits, so for that we do the statistical analysis on the large scale. So lot of statistical studies is required because we are analyzing the gene in the groups or the large level, because analyzing the genome, so analysis of genome, it also involves the comparative genomic study. You can include the comparative genomic study in this. So as I have already told you, this is a part of, this is a method for the pharmacogenomics also, genomic studies. So this method has generated a myriad of robust association for for range of traits. Range of traits means we are considering the different traits of the character. So it can be associated with the same character or the different characters. So we can analyze, that is associated with the disease. Okay, so number, we can have the number of the variants, so we can analyze. So we have different samples, so that is increasing day by day. So this type of study becomes important because this GWAS means genome-wide association studies we carry out on the large scale, means we apply the lot of statistical method for this analysis. And we also use a lot of computational tool in this. Statistical analysis combined with the computational analysis. So you can say genome-wide association studies include statistical analysis and computational analysis. Okay, this is clear to you? Okay, it includes statistical analysis and computational analysis of genomes, genome-wide studies. So GWAS results have a range of applications such as gaining insight into phenotype. So as I have already told you, means we need to make the relationship between genotype and phenotype. For the analysis of the genome, we analyze the relationship between genotypes to its phenotype. So this study includes the biology, estimating heritability, calculating genetic correlations. So this type of analysis study we need to do, making clinical risk predictions, informing drug development programs, and inferring potential causal relationship between risk factors and health outcomes. So Genome-Wide Association Studies aims to identify association of genotypes with phenotypes by testing for differences in the allele frequency of genetic variants between individuals. Means we are taking genomes of different individuals and we are analyzing the association between these two. Okay, can you think of this? Can you read this slide, students? Just wait for a second. So GWAS can consider copy numbers, variants or sequence variation in the human genome. Okay, sequence variation we have already studied and we have okay. So this type of study report blocks of the correlated SNPs. Means we have to make the correlation between the SNPs to its phenotype. Which SNP is associated with this phenotype? Students, do you remember some SNP variation related to the gene therapy that we have discussed in the gene therapy lectures? So there are some variations and mutations that is associated with the disorders. So in the gene therapy lecture we have discussed that. So you can also refer those examples here. But now here what is important, because statistically this is important because we are analyzing the lot of genomes and we are trying to find the SNP's association with the traits and the loci. We are trying to identify the loci which has the genomic risk. Result from GWAS can be used for range of applications. For example, trait-associated genetic variants can be used as control variable in epidemiology studies to account for confounding genetic group differences. Okay, so this is the point. We also consider the epidemiological studies in this to find out the differences among groups, genetic groups. So this is the name of scientist that was given by the Benjamin. Selecting study population, this is very important. We need to select the populations so that we can analyze the variations. So that is very important to identify the group of the, means we need to take the samples. What we want to do, it depends on the interest. Who is the researcher? What is our interest? So it depends on the researcher, scientist, or the group of the people, those who are carrying out the study. Means what is there? Means generally, but this generally the objective behind all these type of studies is to find out the SNP's variation, relationship between the SNP's variation with the genomes, for which the scientist groups work to find out, to identify the cause of the disease. If suppose they are working for a particular disease, in any case, in the case of any disease, so it may be specifically associated with the disease when the scientists are trying to find out the SNP variation associated with that particular disease. So it depends on that. So here the name of the software is given, CAT S14 or GPC 15. So this is so here we can use, okay, we can do some calculation using the software. These are the name of the software. So this is the importance of the computational software, and we can use this software for this study. So one can choose between population based and family based designs. So population based study, that is done on the large scale, and family based designs. Okay, so this study we can say this is at the micro level. A population based study, this is at the micro level. Okay, students, are you getting there may be the difference, because family based designs, family based study, that can be less time consuming, and a population based study that can be more time consuming. But we can take the help of software in this. We take so this is the step, selecting study populations. So on the basis of the resource, it depends on our resources, how much we can do, how much population you can analyze. Can you analyze the population? 1 or 2 students cannot do this. They can choose the family based design or family based study, they can do. But if you want to analyze the population, then we need to work in the group, like you are the group of 103 students. Okay, so it depends, and what type of resources we have, what type of computational facilities we have, because we need to use the software. So on the basis of our resources, so selection of the population is based on that, what is our resources and what we can do. So it depends on that. It is population based study or family based design. The choice of data resource and study design for GWAS depend on the required sample size. I have already told you, it depends on the sample size, whether you are choosing the population or family. So your time or resources is very important. What resources you have? Experimental question, the availability of pre-existing data. What data do you have? Do you have the data of population? Do you have the data of family? Okay, so data is available on the internet or database. You can collect the data from the databases, or you have to conduct the survey or different types of genetic studies to get the data. So you can look on the internet, there are the databases from where you can get the information, and you can analyze that data, what the scientists have already done. You can also carry out that type of studies. So we can look in the databases to collect the information. Already there is a collected information. Okay, like SNP, DB SNP, where we can search in that database for the genetic variation or SNP variation. So that the database, scientists have already done lot of work. So in the bioinformatics, what is our role? Our role is to analyze that data. So we don't need to collect the data. Data is already available. What the others have already done, we need to work on that step. Okay, we don't need to do everything. We can do the computational work and computational study, because already a lot of people in the genetics, researchers in the biotechnology, so they have done the lot of experiment. We can take that data on the NCBI also. So GWAS can be conducted using data from resources such as biobanks. This is the name of one example. Okay, you can search in this, or cohorts with disease focused, means on these resources we will get the information related to disease, population based recruitment, or through direct to consumer studies. Okay, suppose the people are taking medicine, so means we can have the data related to that, what is different kinds of population or family based. So what we do further, then we assemble the data sets. When we have the large data, we make the we organize the data into data sets, so we can analyze that. So there should be sufficient size so that we can run the well powered GWAS. For a complex trait require major investment of time and money, so that may go beyond the capacity of most individual laboratory. So the computational facility should be good, so that we can analyze the large data, because in the genome-wide studies we carry out the different types of analysis. So we should have that facility. However, there are several excellent public resources. So this is useful. Students, you can use those resources. So it provides access to large cohorts with genotypic and phenotypic information. This is important. In the Genome-Wide Association Studies, this is important. What we do in that, we identify a relationship between genotype and phenotype. This is important. Okay, are you getting what is Genome-Wide Association Studies? What is the objective? What we do in that? We make the relationship between genotype and phenotype. Okay. So the majority of GWAS are conducted using these pre-existing resources. What we have means there are so many public resources. Like I told you about the NCBI, you can look for the information. So when new data have been collected in-house, these will typically be co-analyzed. Co-analyze means एक साथ में analysis करते हैं with data from pre-existing resources. We do the comparative study. We get the new data set, then we compare that to with old pre-existing resource. Collecting new data is usually required when more refined phenotype is desired. So we can compare. Then we come to know this is the new phenotype or this is the old phenotype. Whenever we get the new data set, we can compare it to the already existing one. Then we can tell this is the new one or this is the old one. The same thing you do in the sequence alignment. Whenever you get the new sequence, you compare it to the sequences that is already stored in a databases. So this is kind of in the database search we do. So here we do not have sequence. Here we are talking about the variations that is associated with the disease. So we are analyzing the variation. What are the common things and what are the variations? Because we are analyzing genome at the large level. So for all study design, recruitment strategy must be carefully considered. Okay. Collateral bias and other forms of bias in the design means we always try to normalize the data so that there can be no bias in the study. You should not neglect anything. So we should remove the sometimes we also discard some data that can create the bias in the analysis. So next step after that, selecting study population using the computational tool we do. What is the next step? Genotyping of the individual is important. This is done using microarray for common variants. So we have to identify the common variants as well as variants. So common variants means that are common means we are analyzing different population. What are the common variants? If that is the same population, then we can also identify the common variants, and we can also compare the variants with the data of other population from the different data sets. Okay. So the name of the sequencing method WGS include rare variants. So analysis of common variants and rare variants are important. We identify the common variants and rare variants. What we do in the genotyping, when we are analyzing the genotyping and we are identifying the relationship between genotyping and phenotyping. So what is important? Common variants and identification of common variants and rare variants. That is important. So for genotyping, we can use a microarray based method. So you are already familiar of this method, microarray. Genotyping is the most commonly used method for obtaining genotypes for GWAS, owing to the current cost of next generation sequencing. So this thing we can combine with the next generation sequencing. In the NGS data analysis, you have seen how we combine the statistical analysis with the computational studies. So we perform all the integrative method analysis to make the relationship, to analyze the population also. So there we have also seen what we do in the NGS data analysis. So microarray based method is important here. I have told you to listen my lecture on microarray. I have given you that lecture. I have also introduced this technology to you. Okay. I took class on this, and I have also given the lecture video reference for this. So you can attend if you want to know more about the microarray based method. You can attend that lecture to understand role of this technology in the Genome-Wide Association Studies. Okay, so I don't want to go into the detail of microarray methods here. So the choice of the genotyping platform depend on many factors. So that is guided by the purpose of GWAS, means those who are conducting the experiment. The aim is important. Suppose I am conducting the experiment, I am conducting the studies. So I should know though what is my objective. That is guided by that, what we want to do. So all individual cohorts genotyped on the same genotyping platform. Because whenever we are analyzing anything, different things, so we analyze those things on the same platform. Then we can say these are common or rare. We will not analyze the individual, means this one variant with on the other platform, another variant on another. So using the same platform is necessary to analyze all the variants, common variants or different variants. So okay, then we can identify the individual cohorts genotype that have the common things or rare things. So ideally WGS, what is WGS? Whole genome sequence, which determine nearly every genotype of a full genome, is preferred over WES and microarray. Okay, so that is expected to become the method of choice over the next couple of years with the increasing availability of low-cost WGS technologies. Okay, because we are interested in the whole genome sequence, means we are analyzing the genomes. We are analyzing the family genomes or the population genomes. So we prefer to use the WGS sequences. Okay, so this method is useful because in the microarray it has some high cost. Although microarray is very useful, but the cost of this experiment is high. The preparation of microarray, that is very high in comparison to WGS technology, means whole genome sequencing technology. Okay, so although the whole genome sequence is also costly experiment, nowadays it has become easy, means in comparison to the old time, because now we have the advanced technology. So in comparison to microarrays, WGS technology is low cost. So we can carry out a genome-wide association study. So doing that, so it has the significance. It takes a time, the genome-wide association studies, so that are useful because we can reduce the cost of the experiment, because means all the times we cannot perform the microarrays. If you want to go into the details of microarrays, you can attend my lecture that is available on the YouTube. What is microarray? Next step is data processing. So when we have the data, we input the file, ID numbers, coded family relations between individuals. There may be different families. We can have the data of different families, sex, phenotype information. Okay. Covariates, genotypes call for all called variants. So we try to find out, means how they vary. We take the variables, we choose some variable for the analysis like phenotype information. We are making correlation between phenotype to its genotype. So we should know, means which family is getting, whether it is variants, whether it is means We are trying to make the relationship association. What is the association? As the name suggests, Genome-Wide Association Studies means we try to find out the association. First we process that. What we need to process? Deep data processing. What needs? We should have complete information. Means the table and rows, means column and rows. We should have all the information for all the variables that we take in the column, like sex, phenotype information, covariates, genotype cause. Okay, so this is all kind of information we need. Genotyping batch means we have different family genomes, so that is called batch. That is the batch, that is not individual. Following input of the data, generating reliable results from GWAS require means quality controls. We need to be careful because we are carrying out the studies on the large scale. Some examples include steps include removing rare or monographic variants. Okay. Removing variants that are not in Hardy-Weinberg equilibrium. So we are just trying to identify using the Hardy-Weinberg equilibrium analysis. We can select the data. We can remove the rare or monomorphic variants. We can remove the variants that are not in Hardy-Weinberg equilibrium. So we can filter the SNPs that are missing from fraction of individuals in the cohort, identifying and removing genotyping errors, and ensuring that the phenotypes are well matched with the genetic data. So we just trying to remove the error by comparing self-reported sex versus sex based on the X and Y chromosome. So for the analysis of, we can identify the data related to means male or female or X and Y chromosome. Okay, are you getting, students? Are you here? I think there is a loss of connection. Hello? Hello?
Yes, ma'am.
Okay, when there was this disconnection, I think there was disconnection.
Yes, ma'am.
Okay, there was the internet was disconnected. Okay, before that. Are you listening the lecture? Are you listening or not?
Yes, ma'am. Yes, ma'am.
Okay. Okay. Just the lecture is on the recording. If there was a disconnection, you will get that. Okay. So yeah. Okay. So I was discussing this step, data processing. We need the information of variable like phenotype information, covariate, genotype, cause. Okay. We need to analyze. So it depends what we want to do. That all we do in the batch and what is our objective. Okay, we just test the variants whether they are following up the Hardy-Weinberg law or equilibrium or not, and we can filter the SNPs that are missing from the fractions of individual. Okay, so in that way we can remove the identify the genotyping errors or remove that. Okay, and we can also distinguish between the X and Y chromosome data or the sex, male or female data. So software tools we can use, this is the name of Plink is one tool, have been specifically designed to analyze genetic data and can be used to conduct many of these quality control steps 20. So the name of software, we can use this software for the quality control analysis. Table is not given here, means that is this I have taken from the research paper. I will send you the research paper also, review article. You can view. Once the sample and variant quality control have been performed on the GWAS array data, variants usually undergo phasing. So then we can we can process the data using principal component analysis, and we can identify all the individuals to define the clusters. Cluster means group of the individuals data or that have the similar genotype. Why it is done? To identify the common variants. Means they are the variants. Although we are identifying the variant, but variants are clustered according to the similarities and differences. Similar genotypes, we can group them in the one cluster. As this is done first to identify and exclude outliers. We exclude the outlier, means the genotypes that are not showing the similarity. So we can exclude by the statistical analysis. We can exclude the outliers. Okay, so the range analysis or the interquartile analysis, that type of different studies can be used to identify the outliers. Statistical method can be used here. So then to compute and include the principal components as covariates in the subsequent GWAS regression model. Means what? Regression method we can also use that. I mean principal component analysis is the one method, and regression studies we can carry out. A linear regression is one of the important method in this. So we can analyze the deviation between the different groups. Means how they are deviating from each other. Means we can set the criteria of similarity. Means we can draw the line. We see that regression. In the microarray lecture, I have told about the regression method. There is a line, means if there are the different clusters. So if data is scattered, the population data is scattered, there is no correlation among the x and y variable. If there are two variables and we have no correlation among these two, then data is, we get that in the scattered form. So sometimes there is a correlation, like the plant height depend on the sunlight. Okay, so there is a positive correlation. Sometimes there is a negative correlation, like the कुछ पेड़ पौधे होता है ना जो कि सनलाइट में नहीं ग्रो करता है, तो देयर मे बी द नेगेटिव कोरिलेशन आल्सो. सो that type of study is called correlation studies. But if there is no correlation among x and y variable, then we opt for the regression methods when data is scattered. Because we are so here it is important, we are analyzing the variants, common variants, and we are grouping them together. We are making clusters. We are trying to make the groups of those. So all the variants are different, but we are trying to identify the common groups of the variant. Although they vary from each other, but still we are trying to find out the, means there is no relation between x and y, but we are trying to find out the group. So how we will do that? Okay, so you can think means regression model, इसमें क्या होता है, क्लस्टरिंग में क्या करते हैं, क्लस्टरिंग भी एक मेथड है. You can think if there is the two different groups, okay, like people are sitting in the same room, I can explain you like that, and there is one group is sitting in the one corner, and another group is sitting on the other side. There are two sides, left hand side and right hand side. Now how can you make the clusters or group of those individuals on the left hand side and right hand side? How can you say these are the, how many groups are there? So what you can do, to make the groups of when there is no relation between these individuals. The peoples are sitting, like peoples are sitting on the railway stations, so they don't have any relation, but they all go there to catch their train. Okay, they are waiting for that. So what is the common thing in them? They are waiting for their train. So we can group them कि किस तरह से हम ग्रुप करते हैं. तो सेम रूम में अगर कुछ लोग बैठे हैं, हम कैसे करेंगे जबकि वो अलग अलग हैं. ऑल आर डिफरेंट, बट वी आर ट्राइंग टू फाइंड आउट द कॉमन थिंग्स अमंग. तो कैसे हम उन्हें ग्रुप करेंगे? कोई बताएगा तरीका? Any one of you can tell me? If the different peoples are sitting in the same room, how can you cluster them together? How can you group them together? Can you tell me the way? You can identify their common interest. What is their common? क्या है उनका कॉमन इंटरेस्ट, क्या होता है? ठीक है, बट अगर हम यहाँ डिस्टेंस लेते हैं, इफ वी आर ग्रुपिंग द पीपल्स ऑन द बेसिस ऑफ डिस्टेंस, सो वी कैन ड्रॉ ए लाइन इन द बिटवीन. कहीं पे हम एक लाइन ड्रॉ कर लेंगे, ठीक है, डायगोनल लाइन हमने ड्रॉ कर ली सपोज, और उस लाइन पे खड़े होके हम देख रहे हैं कि ये जो राइट हैंड साइड में है वो कितने दूर हैं इस लाइन से, और जो लेफ्ट हैंड साइड में है वो कितने दूर हैं, डिस्टेंस कितना है. इफ दे हैव द सेम डिस्टेंस वी कैन ग्रुप दैम टुगेदर. इफ दे मैं बीच में हूँ, खड़ी हूँ, और उसके इस तरफ कितना डिस्टेंस है और इस तरफ, वी कैन ग्रुप दैम टुगेदर if the peoples are on the same distance from that line. So in that way we can minimize the deviation distance between them. जो कि 10 मीटर का या 10 फीट का डिस्टेंस है, उसको हम कह सकते हैं कि उस लाइन से वो ये इंडिविजुअल 5 फीट के डिस्टेंस पे है, और जो राइट हैंड साइड में है वो भी 5 फीट के डिस्टेंस पे है. तो सेम लाइन से लेफ्ट हैंड साइड में जो है वो भी 5 फीट के डिस्टेंस पे खड़ा है, और उसी सेम लाइन से मीन्स the peoples are sitting there at the same distance on the right hand side. So 5. So now we can characterize that A and B into same group. Okay, are you getting what I am saying? Are you all getting what I am saying? Yes, tell me. है ना, दैट इज द मीन्स इन द स्कैटर्ड डेटा व्हाट वी डू. स्कैटर्ड प्लॉट हम क्यों करते हैं? जिनके एक्स और वाई में कोई रिलेशन नहीं होता, बट स्टिल वी आर ग्रुपिंग दैम टुगेदर. सो द इन दैट वे वी यूज द रिग्रेशन मॉडल. सो सबसे आसान तरीका है कि हमने एक लाइन ड्रॉ कर ली और फिर इस लाइन से हम डेविएशन देखते हैं. दैट इज ऑल्सो कॉल्ड कर्व फिटिंग भी कहते हैं. यू कैन अटेंड माई यूट्यूब लेक्चर ऑल्सो फॉर द मोर डिटेल. क्लस्टरिंग भी मैं आपको आई विल एक्सप्लेन यू. I can explain you in the another class also. So here I am saying the clustering हम क्या करते हैं. एक तो क्लास होता है कि आपकी क्लास है, that is based on the your roll number. आपका रोल नंबर है 1 to 120, तो हम ये कहेंगे कि this is the same class. Now there can be the another way of grouping you together. In the clustering we don't have the predefined criteria. Now you are the student of B.Sc Biotechnology, so this is the predefined criteria because you have chosen this subject biotechnology. So that is the classification method. इसे हम कहते हैं classification. But clustering is different from classification. How it is different? I can make you the different groups. I can divide you in the different groups on the basis of what? अभी मैंने क्या किया था, रोल नंबर के अकॉर्डिंग आपके ग्रुप्स बनाए थे ना, प्रैक्टिकल ग्रुप्स. So that is the classification way. Classification means predefined information I have used. What is the predefined information? Your roll number I have used. That method is called classification. Now what is clustering? If I divide you on the basis of some different criteria, like on the basis of your interest, I can assign you some seminar topics. I can make the different groups on the basis of your interest. Means what type of research project you want to do, what type of seminar you want to do, what type of, okay, somebody will be interested in the gene analysis, somebody will be interested in the protein analysis, protein structure prediction, protein modeling, and some people may be interested in the gene analysis or genome analysis. So I can divide the on the basis of your interest. ओके लेकिन ये मुझे पहले से नहीं पता होगा ना. आई विल गिव यू द चॉइस या कि आप विच वन यू विल चूज. सो आई डोंट नो. प्री डिफाइंड नहीं है. क्लस्टरिंग में क्या है, कुछ भी प्री डिफाइंड नहीं है. इट डिपेंड्स ऑन योर चॉइस विच टॉपिक यू आर गोइंग टू चूज. इट्स अप टू यू. आई कैन नॉट डिसाइड इन द प्रायर. सो रोल नंबर इज डिसाइडेड. तो वो क्लासिफिकेशन मेथड हो गया. लेकिन क्लस्टरिंग क्या है कि इट इज बेस्ड ऑन यू व्हाट द पीपल्स विल चूज, व्हाट यू आर गोइंग टू चूज. और जो जितने भी लोग जीनोम एनालिसिस चूज करेंगे जीनोम ग्रुप में आ जाएंगे. जीनोम एंड हाउ मीन्स द अदर स्टूडेंट कैन चूज द प्रोटीन स्ट्रक्चर प्रेडिक्शन. सो देयर कैन बी 2 ग्रुप्स अकॉर्डिंग टू दैट. अभी मुझे पहले से नहीं पता है ना कितने ये चूज करेंगे कितना. ठीक है. आर यू गेटिंग व्हाट आई एम सेइंग? आर यू गेटिंग व्हाट आई एम सेइंग? जस्ट टेल मी. Nobody is here to reply. Why you are not replying? Are you there? So clustering we can make the means it depends कि how the means जो भी लोग हैं उनमें क्या है ये निकल ऐसे. In that way we are trying to identify the common variants although they are different. सो इट डिपेंड्स कि उनमें कुछ सिमिलैरिटी है तो वो निकल के आएगी. लेकिन पहले से नहीं पता होता है ना ये. सो दैट्स व्हाई वी आर कैरिंग आउट द जीनोम वाइड एसोसिएशन स्टडीज. दिस विल बी रिवील्ड आफ्टर दैट. अब जैसे मैं आपको कोई चॉइस फॉर्म दूंगी तो उससे मुझे रिवील होगा कि कितने क्लस्टर बनेंगे और कितने हाउ मेनी ग्रुप्स विल बी देयर ऑन द बेसिस ऑफ योर इंटरेस्ट. आया समझ में? देयर कैन बी यू कैन गिव some people can give their different interests, so there can be 3 groups 4 group on the basis of the student interest. So in the class also you can make the different groups on the basis of your interest. So that is the clustering. ठीक है. मैं आपको कहती हूँ कि यू डू द स्टडी फॉर रोल नंबर 1 to 15 एक साथ बैठ के स्टडी कीजिए. फिर 16 से 30 तक दूसरा ग्रुप हो गया. बट ऑन द बेसिस ऑफ योर इंटरेस्ट यू कैन मेक योर ओन ग्रुप. So that is revealed after when you carry out that study, that will be revealed at that time. उसके पहले नहीं पता होगा कि कितने ग्रुप बनेंगे. So that is the way how we do the clustering. There is a difference between clustering and classification. I have explained you. So what we why we use the regression models. So testing for the association. So association study is also important. What is the association between different things? Okay. So typically GWAS. So linear or logistic regression models are used to test for the association. अभी मैंने आपको explain किया why we use the regression method when there is no correlation. डिफरेंस है उनमें और उनमें डिस्टेंस है तो फिर भी हम उनके एक ग्रुप बनाएंगे. क्लस्टरिंग करते हैं फिर भी डिपेंडिंग ऑन वेदर द फिनोटाइप इज कंटिन्यूअस. हाइट तो ये है. दीज आर द वेरिएबल्स. हाइट, ब्लड प्रेशर, बॉडी मास इंडेक्स. यू नो ब्लड प्रेशर एक अलग चीज़ है, वेरिएबल है. हाइट एक अलग चीज़ है. लेकिन फिर भी हम इसका कोरिलेशन निकालते हैं. वी आर ट्राइंग टू फाइंड आउट द कोरिलेशन. मीन्स इस तरह से कि वो किस तरह से वो रिलेटेड हैं. ग्रुप बनाएंगे. बॉडी मास इंडेक्स. यू नो दीज आर डिफरेंट थिंग्स. हाइट और ब्लड प्रेशर में कोई रिलेशन नहीं होता है. बट वी आर डूइंग द ओके देयर कैन बी. लेकिन ये तब पता चलेगा जब एनालिसिस करेंगे. पहले से नहीं पता होता है ना हमें. हाइट, ब्लड प्रेशर, बॉडी मास. तो वी कैन एनालाइज द डिफरेंट पॉपुलेशन एंड वी कैन टेल कि हम बच्चों का एनालिसिस करेंगे चाइल्ड ग्रुप, देन वी विल एनालाइज द एडल्ट ग्रुप. तब हम ये बता पाएंगे कि कितने ग्रुप निकल के आते हैं. आर यू गेटिंग? आर यू गेटिंग स्टूडेंट्स? हाँ, पहले से नहीं पता होगा ना. जैसे 100 है, अब कितने किस ग्रुप में कितने ग्रुप बनेंगे ये हमें नहीं पता होता. क्लासिफिकेशन मेथड, इन क्लासिफिकेशन मेथड वी हैव प्री डिफाइंड इन्फॉर्मेशन बट हियर वी डू नॉट हैव प्री डिफाइंड. अगर पहले से स्टडी हो चुकी है तो प्री डिफाइंड है. जैसे कि आपके रोल नंबर असाइन कर दिए गए बिकॉज यू आर द स्टूडेंट ऑफ B.Sc बायोटेक्नोलॉजी. बट हियर इट कैन बी वेरिड. वी आर कैरिंग आउट द जीनोम वाइड एसोसिएशन स्टडीज. सो ओके, एसोसिएशन हम निकालेंगे. क्या एसोसिएशन है? ऐसे मार्केट में हम एनालिसिस करते हैं एसोसिएशन. जैसे ना somebody is purchasing somebody is purchasing bread, तो हम क्या सोचते हैं, वो शॉपकीपर क्या करता है कि चलो बटर रख लेते हैं, जो ब्रेड खरीदेगा आमिस वो बटर भी खरीद सकता है. Is it true? तो हम रख सकते हैं. सो दिस इज कॉल्ड मार्केट एनालिसिस करते हैं इसको. तो वो भी एक काइंड एसोसिएशन है कि हम एसोसिएशन एनालिसिस करते हैं, लिंक एनालिसिस, क्या लिंक है आपस में, परफॉर्म करते हैं. सो सेम थिंग, सेम टेक्निक्स वी अप्लाइड हियर इन द एनालिसिस ऑफ द population. I will give you my recorded lecture on the association. Okay, I have one recorded lecture. I will just share with you on the drive. Okay, you just attend. What is association or some statistical term, क्या है, ठीक है. Then you will understand. Then you can apply that anywhere. कहीं भी आप उसको apply कर लेंगे. This is not only this case. Here we are using that for the Genome-Wide Association Study. So covariates such as age, sex and ancestry are included to account for a stratification. Stratification means different levels हो जाते हैं जब. And avoid confounding effects from demographic factors. ठीक है, पॉपुलेशन डेटा एनालिसिस हम करते हैं तो उसके कौन से फैक्टर है जो अफेक्ट करते हैं, ये हम एनालिसिस करते हैं विद द कैविएट. कैविएट मीन्स ग्रुप, स्कैफोल्ड, ग्रुपिंग करना. सो दैट दिस में रिड्यूस स्टैटिस्टिकल पावर फॉर बाइनरी ट्रेस इन अ सर्टेन सैम्पल. व्हाट इज रिटन मीन्स if we will characterize our data according to this, so statistical power will be reduced, means we need to do then less statistical analysis, but we have to carry out the means we are only carrying out the statistical analysis for large data sets. So we can set some criteria and we can remove. Okay, कि which data we should discard from the analysis. If it doesn't fit for my interest, we can discard that data. It depends what I am going to do. If I am looking for the data for the one disease that is not related to my interest, I can discard that data. This is true. It depends what you are going to do. You go to the market, you are going to purchase it, so you can discard and you can choose it based on your interest what you want to do. Okay, so it depends कि जो भी रिसर्च कर रहे हैं, एनालिसिस कर रहे हैं, उनका इंटरेस्ट क्या, किस डिजीज में है इंटरेस्ट, क्या एनालिसिस करना चाहता है. ऑन द बेसिस ऑफ दैट वी कैन डिस्कार्ड. ठीक है. अब ह्यूमन जीनोम प्रोजेक्ट में ऐसा नहीं था. इन द ह्यूमन जीनोम प्रोजेक्ट वी हैव कंपेयर्ड एवरीथिंग. दैट वाज द लार्ज कॉस्ट, मीन्स द एक्सपेरिमेंटल कॉस्ट हाई थी और हमने किया था क्योंकि हमने एक बार में सब कुछ करना था. ठीक है, that's true. But in the Genome-Wide Association Studies we can have certain interest, डिजीज से रिलेटेड है जो भी है. बट इन द एक बार हमने कर लिया है सब कुछ ना, तो सारा फिर कुछ नहीं करेंगे. वी विल नॉट रिपीट इट अगेन. ह्यूमन जीनोम प्रोजेक्ट वी हैव ऑलरेडी इन्वेस्टेड, वी हैव ऑलरेडी डन सो मेनी थिंग्स, सो व्हाई वी आर गोइंग टू रिपीट इट अगेन. Statistical analysis we can perform using that data. Okay, because we have the genome, we have done the comparison. Now what we are looking for, we are looking for the SNPs, or we are identifying the common variants and rare variants. So we will discard, we can discard some data if it doesn't suit to my interest. So we can improve the statistical power, like if we use the method like association. Association means जैसे शॉपकीपर है वैसे ही सामान रखता है. अब उसने नई नई शॉप खोली है, वो कैसे सामान रखेगा कि एक पर्सन यहाँ पे ये परचेज करने आता है ब्रेड, तो वो बटर भी परचेज कर लेगा. ही विल कीप बटर फॉर द नेक्स्ट डे. तो वो क्या करता है, किस लिए करता है वो, दैट इज टू इम्प्रूव द सेल, इसलिए करता है वो. सो इट्स अप टू हमें क्या चाहिए, व्हाट इज माई इंटरेस्ट, ये है. और हम देखेंगे क्या आ रहा है. इनिशियली फर्स्ट डे वो जैसे शॉप शुरू करता है, उसे नहीं पता होता ना यहाँ की पॉपुलेशन क्या खरीदेगी, क्या परचेज करना है. तो जब उसे धीरे धीरे प्रैक्टिस होती है, व्हेन पीपल्स आर डिमांडिंग समथिंग, तो ही विल, शॉपकीपर विल कीप दैट थिंग. और एक चीज़ वो रखेगा तो साथ में दूसरा सजेस्ट कर सकता है कि मेरे पास आई हैव दिस, आई हैव बटर ऑल्सो, डू यू वॉन्ट दिस, इस तरह हो सकता है. ठीक है, दिस इज क्लियर टू यू स्टूडेंट्स. That is clear to you?
Yes, ma'am.
Okay. So in that way we can improve our analysis. Generally, statistical computation methods are common. That is applied here. So the same method. I have the another lecture on the association. You can attend that. Method we should clear that we can use anywhere. So because here we are analyzing the large genomes of the different population. That is correct. What we are doing here? We are identifying the common variants and rare variants. और हम क्या कर रहे हैं इसमें जीनोम वाइड एसोसिएशन स्टडीज में जस्ट टेल मी वी आर ट्राइंग टू आइडेंटिफाइंग रिलेशनशिप बिटवीन जीनोटाइप एंड फिनोटाइप दैट इज इंपॉर्टेंट पॉइंट इसको दैट यू रिमेंबर दिस ओके अब वी आर यूजिंग द स्टैटिस्टिकल मेथड एंड कंप्यूटेशनल मेथड्स ठीक है ये आपके दीज आर द रेफरेंसेस इंपॉर्टेंट रेफरेंसेस आई हैव प्रिपेयर्ड दिस लेक्चर यूजिंग दीज रेफरेंसेस ओके. So if you want the details, I will send you these papers also, and if this is sufficient, you can, okay, this is sufficient, this slide is, you can read this also, okay. So now we will see, I want to show the figure, actually, sorry, this is, okay. This is the Hugo. I also have, sorry, I want to show you. This is the lecture. I have also shown that figure. I have not pasted because this is a large figure. We have discussed this figure like data we collect from different individuals or population. This can be family based or population based. There can be two types of analysis, that is family or population. So family analysis will be done on the short scale. This is large population होगी तो ज़्यादा टाइम लगेगा. जीनोटाइपिंग करते हैं फिर हम. So we have discussed these steps. Then we do the quality control analysis. Means this is the data from the different populations. Here you can see, okay. Then we carry out the principal component analysis or association analysis. Different types of analysis we do. So here this is the result of principal component analysis. So here you can do the clustering of this. Are you able to see this, students? This
मैम स्लाइड शो नहीं हो रही है.
नहीं हो रहा है. Why?
यस मैम कुछ शो नहीं हो रहा.
So अभी लेक्चर में क्या हो रहा था फिर अभी यू वर नॉट एबल टू सी.
मैम सिर्फ वॉइस आ रही है, स्लाइड बहुत देर से शो नहीं हो रही है.
एक मिनट तो यू शुड हैव टोल्ड ना. व्हाई ओके यू आर लिसनिंग फॉर सम टाइम व्हेन देयर वाज डिस्कनेक्शन, सो यू शुड हैव टोल्ड मी. How I will come to know when there was a disconnection at the middle. So after that it is not okay. Here you can see I have shown you this. I have already shown you this figure on the last turn. Okay. Switch off your mic. So we collect the data. This is the first step. Then we do the genotyping. Okay. Then we do the quality control analysis. Okay. Principal component. So this is the result. Are you able to see?
Yes ma'am.
African आपका डेटा यहाँ पे Asian. This is the clustering. This is the way of cluster. How we do the clustering है ना. This is the African. So एक हम कह सकते हैं कि जो African population, the people who are belong to the African, they will come in the one cluster. Okay. The people who belong to the genome, who belong to the American population, they will be clustered in one. ठीक है. तो ये एक प्री डिफाइंड भी हो सकता है इसमें कुछ चीज़ें. पर हियर इन द लाइक हम क्लस्टरिंग करते हैं उसमें हमें नहीं पता होता है ना जो हम कर रहे हैं. मीन्स व्हाट वेरिएबल्स वी हैव चूजन. लाइक इट डिपेंड्स कि हम जो ग्रुपिंग कर रहे हैं इन द क्लस्टरिंग मीन्स विच वेरिएबल वी हैव टेकन, व्हाट जीनोटाइप और फिनोटाइप वी हैव टेकन. इट डिपेंड्स ऑन दैट. that will be done on that. तो clustering में हम ये करते हैं. Association, what we, after the principal component analysis, we perform the SNP. We analyze the SNP's relation to the genotype. Genotype we already know. We have already done the genotyping. Then we are trying to make the correlation. These are the SNPs. SNP 1, 2, 3, 4, 5, 6. So here we can see on the same position the person has G, then C. So in the different person we have the different. So these are the common variant. Person 1 and 2, you can say SNP 1 is common in this. Are you able to see? And person 3 and person 4. So here for SNP 1 we can make the two group of this. Are you able to see?
Yes, ma'am.
We can make the two group because these person 1 and 2 has guanine on SNP 1. ठीक है? Okay. So person 3 and person 4 we can make the another. So within the same SNP we have the two groups. We can have the two variant of the same SNP. Now look at the SNP 2. So T and T. Person 1 has same person 2. So here also this thing is common, person 1 and 2. So person 1. So in the if we have analyzed the two SNPs, so these have the common thing. So these are the different group. Person 1 and 2 can be grouped in the 1 and person 3 and 4 can be grouped in 2. So that's not only true in these two SNP variation. We can look at the another SNPs also. We can count how many times it is in. So here you can see in SNP 3 G is similar. So person 1, C. So now there is a variation here. Okay. Now in the SNP 3, when we are grouping SNP 1 and SNP 3, so now what is common thing? So G and G is common here. Person 1 is common, is SNP 1 and SNP 3 means we have the common variation here, SNP variation. So we can group. अब इसकी grouping हम किससे करेंगे? Now person 1 is sharing relationship with this SNP 3. इस position पे G आ रहा है यहाँ person 3 में. Now person 1 can be grouped with person 3 when we analyze the SNP variation 3, because this may be associated with the different phenotype. So it depends on the phenotype and genotype. Are you getting what I am saying? Are you getting?
Yes, ma'am.
Now person 1 means has G here at this position when we are analyzing SNP 3, and person 3 is also having G, and person 2 is having C and 4. अब grouping किसकी हुई? person 2 and 4, and person 1 and 3 has one group. So when it is associated with SNP 3, so SNP 3 variation will be associated, इसका association किसी और genotype phenotype से होगा. ठीक है? Okay, this is clear to you? This is clear to you? Same thing you can say here SNP 5 when we are looking the SNP variation 5. So person 1 and 3. So for SNP 3 now, so for SNP 5, person 1 and person 3 can be grouped together, and person 2 and 4 can be grouped together in another group in case of SNP 5. This is true? This is true? Just tell me. This is the similar case as in SNP 3. ये common variants हो गए ना. Person 1 and 3 की grouping किस में हो रही है? In case of SNP 3, G is similar. Person 1 and person 3, G similarity हो रही है यहाँ पे. C की grouping हो रही है. Person 2 and 4 की grouping हो रही है. Same thing is happening here in case of SNP 5. So we can say SNP 3 and SNP 5 are common variants. Are you getting?
Yes, ma'am.
SNP 3 and SNP 5 are common variants here, or we can identify the rare variants. Okay, on the basis of that. Now it is clear how we can do this. So this is the example. Now you just explain me SNP 3. I am giving you this exercise. You just explain. SNP 6. How you can group? Tell me the group. Just tell me. In case of SNP 6.
Ma'am, person 2 and person 4.
Okay. Person 2 and person 4. Why person 3 is also common, na? C or C. So in the 3, person 2, person 3 and person 4, we can group them together in one group. Is it true?
Yes, ma'am.
A person 1 has the, so this is the rare variant. Okay, here. Okay. Person 1. Okay.
Ma'am.
Okay. So now it is clear?
Yes, ma'am.
Okay. So association testing is here. So I have already told you what is association event. So same thing you can apply here. Now you consider we have genome where, because we are analyzing genome and variation. So SNP variation population. I have explained you using the example of bread and butter, but you can apply that concept here. Okay. Which genotype is associated with which phenotype? Okay. On the basis of that. Here you are saying different SNPs are associated, obviously that will be associated with the different genotypes and phenotypes. Okay. So on the basis of person groupings or they are related to their data, you can compare using the variables data. So it can be compared here. What is the association? Okay. So chromosomes. So chromosomes are depicted here 1, 2, 3, 5, 6. So here we can say ये वेरिएशन आ रहा है तो हम कह सकते हैं ना यहाँ पे कि यहाँ पे है, कहाँ है ये एसोसिएशन क्या है, उसकी एसोसिएशन क्या है, क्रोमोसोम 17 और इसके केस में यहाँ तक जा रहा है. ठीक है. सो लॉगरिथमिक एनालिसिस किया है हमने, यहाँ पे प्रोबेबिलिटी ली है. सो एनालिसिस करके हम जान सकते हैं कि व्हाट इज द एसोसिएशन, क्रोमोसोमल एसोसिएशन ऑफ दीज वेरिएशंस, एसएनपी वेरिएशंस. ठीक है. ओके. एसोसिएशन हम निकाल सकते हैं कि विच क्रोमोसोम मीन्स ये दीज एसएनपी वेरिएशन आर एसोसिएटेड विद विच क्रोमोसोम्स, उसकी लोकेशन क्या है, लोकेशन भी हम जान सकते हैं कि क्या होती है. जैसे कि आई एम टेलिंग यू वन केस लाइक क्रोमोसोम 23 और 9 इज रिलेटेड विद ईच अदर इन केस ऑफ CML, ल्यूकेमिया डिजीज होती है. ठीक है. उसमें क्या होता है, CML फॉर्म में क्या होता है, क्रोमोसोम 9 इज एसोसिएटेड विद द क्रोमोसोम नंबर 23 से एसोसिएट हो जाता है वो. ठीक है. तो एक डिजीज होती है CML, तो वो एक उसमें ये चेंज आता है. ठीक है. सो हियर आई एम टॉकिंग अबाउट द, बट वी आर टॉकिंग अबाउट द SNP वेरिएशंस. ये आपका है और उसके बेसिस पे हम फिर आगे फर्दर वी डू द एसोसिएशन एनालिसिस. व्हाट इज द एसोसिएशन बिटवीन जीनोटाइप एंड फिनोटाइप्स मीन्स we can do here. Okay. And then we do the meta analysis, we do the more refined analysis using that data, using the association data, principal component analysis data. We further analyze that data, for that is called meta analysis. We are applying the lot of methods. Meta data analysis हम कर रहे हैं, we are applying the multiple methods and we are also taking the large set for the analysis. But now we have refined data of different things, genotype, phenotype, SNPs and okay, we have different kinds of data like principal component analysis data and association analysis data. So now we have different analysis. Okay, is it clear to you? क्योंकि हमने ऊपर क्या किया, we have done the genotypic analysis, association analysis, इस इतने तरह का हमारे पास हो गया ना डेटा. So now at the cohort, okay, cohort. So now what we call them, cohort A, cohort B, cohort C, मीन्स डिफरेंट एनालिसिस करके हमने डेटा को कंबाइन किया है और उसके बेसिस पे हमारी ग्रुपिंग बनी है, कोहॉर्ट A, कोहॉर्ट B, कोहॉर्ट C. नाउ वी हैव डिफरेंट ग्रुप्स. ओके, आर यू एबल टू सी? सो देन वी डू द रेप्लिकेशन एनालिसिस, कि उसकी कॉपी हम बनाते हैं. रेप्लिका बनाते हैं, कॉपीज बनाएंगे और उसको एनालिसिस करते हैं, ठीक है। देन हम करते हैं पोस्ट जीवास एनालिसिस। आफ्टर दैट उसके बाद में हमारा डेटा और भी रिफाइन हो जाता है। वी अप्लाई द सो वी कंबाइंड, ये आपकी क्लस्टरिंग हो रही है। हियर यू कैन सी ना, वी आर डूइंग द क्लस्टरिंग, मीन डिस्टेंस देख रहे हैं इसका इससे कितना डिस्टेंस है, व्हाट इज द डिस्टेंस बिटवीन दिस नोड टू दिस नोड, व्हाट इज द डिस्टेंस बिटवीन दिस नोड टू दिस नोड, ये डिस्टेंस फाइंड आउट कर रहे हैं। और इस बेसिस पे, आर यू गेटिंग व्हाट आई एम सेइंग, इज इट क्लियर टू यू, इज इट क्लियर टू यू?
नो मैम, एक बार और बता दीजिए।
हाँ, डिस्टेंस मीन्स मैंने आपको एग्जाम्पल दिया था ना रिग्रेशन में, क्लस्टरिंग हम कर रहे हैं, तो ये दिस इज द क्लस्टरिंग। और ये डिस्टेंस किस बेसिस पे निकल के आया, इसको आप ऐसे डिस्टेंस मान लो जैसे हम रूम में खड़े हैं ना, लाइन ड्रॉ कर ली फिर वहां से डिस्टेंस, लेकिन ये लीनियर रिग्रेशन नहीं, ये है कि जैसे दो चीजें हैं, दो जीनोम है, उनके बीच में कितना डिस्टेंस है, दो SNP कितना डिस्टेंस है, कैसे पता चलेगा डिस्टेंस। हियर इन SNP क्या किया था आपने, यहाँ पे क्या किया ना, कितना डिस्टेंस है यहाँ पे, SNP 6 और सॉरी ये SNP 3 है, दिस इज SNP 3 और ये किसके सिमिलर है, SNP 5। हम कह सकते हैं इनमें सेम डिफरेंस है। देखिए SNP 3 एक डिफरेंट वेरिएशन है, SNP 5 इज डिफरेंट वेरिएशन, लेकिन कॉमन क्या है यहाँ पे, person 1 is grouping with person 3, okay, and person 2 is grouping with person 4. This is the common thing between SNP 3 and SNP 5. Are you able to understand? Because grouping is similar here, person 1 is making group with person 3, person 2 is making group with person 4. This is the similarity amongst SNP variation 3 and SNP variation 5. Although there is no relation between these mutations, ठीक है। तो ये इनका ग्रुप, ये ग्रुपिंग जो है ये अलग तरह की ग्रुपिंग हो गई ना, कॉमन वेरिएंट्स हो गए। आर यू गेटिंग भाई, ये म्यूटेशन अलग है SNP 3 में भी SNP 5 में भी, लेकिन ग्रुप सेम है ना, पर्सन 1 का 3 के साथ ग्रुप बन रहा है दोनों में, और पर्सन 2 इज ग्रुपिंग विद द पर्सन 4 in both the variation. This is the common thing, this is the clustering method, what I am trying to say, ये मैं आपको एक्सप्लेन कर रही हूँ अभी। तो अब यहाँ पे हम क्या देखेंगे, डिस्टेंस देखेंगे कितना रेजिड्यू। अब लास्ट वाले में क्या था, हियर SNP 6 पर्सन 2 3 4 आर ग्रुपिंग टुगेदर 1, और ये आपका वेरिएंस है, अलग हो गया ना, तो ये 3 जो है वो क्लोज हो गए। अब डिस्टेंस हम देखेंगे यहाँ पे कि node 1 is in terms of variables. Now how the variables show the relation with each other, means association. Now we have already performed the association analysis, already performed. So now we can group them together on the basis of clustering. अब हम clustering कर सकते हैं ना, जैसे मैंने आपको एग्जाम्पल दिया ना आपकी क्लास में, on the basis of your choice I can make your different groups, ठीक है। तो जब हम स्टार्टिंग करते हैं रोल नंबर वाइज ग्रुप बनाते हैं वो क्लासिफिकेशन, लेकिन हियर वी आर डूइंग द क्लस्टरिंग। कैसे कर रहे हैं, देखिए पहले तो हमने पॉपुलेशन वाइज ग्रुप बनाए थे ना, अफ्रीकन निग्रोज और ये सारे, दैट इज प्री डिफाइंड, वो क्लासिफिकेशन प्री डिफाइंड हो गया ना वो। नाउ हियर दिस इज ऑन द बेसिस ऑफ द रिजल्ट्स, अभी क्या निकल के आया उसके बेसिस पे है पोस्ट जीवास एनालिसिस। सो हियर वी विल सी कि कितना डिस्टेंस है, अब किसके बीच में कितना डिस्टेंस है, दिस इज बेस्ड ऑन योर एनालिसिस, व्हाट जीनोटाइप एंड फिनोटाइपिक एनालिसिस यू आर डूइंग, दिस इज ऑन द बेसिस ऑफ दैट, दिस इज नॉट ऑन द बेसिस ऑफ अफ्रीकन निग्रोज एंड डिफरेंट टाइप्स ऑफ पॉपुलेशन। अब ये जो आपने वेरिएबल लिए थे ना, यू आर ट्राइंग टू फाइंड आउट द रिलेशनशिप बिटवीन जीनोटाइप टू फिनोटाइप, एंड व्हाट इज द एसोसिएशन बिटवीन द डिफरेंट वेरिएबल्स कि हाइट, बॉडी मास, ब्लड प्रेशर, क्या रिलेशन है। नाउ इट विल बी डिपिक्टेड बाय द क्लस्टरिंग मेथड, क्लस्टर्स। यहाँ पे आप देखो ये जो 4 हैं ये पास पास आए तो इसको हम एक क्लस्टर में रख सकते हैं, और अगर ये इससे दूर है तो इसको हम डिफरेंट रखेंगे ना, जो पास पास है वो सेम क्लस्टर हो जाएगा, जिनका आपस में एसोसिएशन निकल के आएगा वो सेम क्लस्टर में होंगे। नाउ इन दिस टेबल यहाँ पे आप कलर के बेसिस पे देख सकते हैं, द ये दिया है ना स्पॉट मीन्स जिसकी कलर ज़्यादा है, कलर इंटेंसिटी, तो इसको हम कह सकते हैं कि इसमें ज़्यादा एसोसिएशन है, ग्रुपिंग हो रही है, सेम है उनका। ठीक है, डिफरेंट कलर में है तो वो डिफरेंट है, सेम कलर वाले। अब यहाँ पे अब देख लो कितना है, ये ग्रे कलर, लाइट ग्रे, ओके, लाइट ब्लू, डार्क ब्लू, तो ऑब्वियसली जो ब्लू ब्लू कलर वाले वो सेम ग्रुप होगा ना, क्यों? जैसे कि हाई ब्लड प्रेशर है, जैसे ब्लड प्रेशर है ऑब्वियसली, तो ये लो वाला, ये वाला तो कम और ज़्यादा ब्लड प्रेशर हुआ ना, तो कम, लो ब्लड प्रेशर, हाई ब्लड प्रेशर, तो ये डेटा एक उससे रिलेटेड हो गया ना फिर। आर यू गेटिंग व्हाट आई एम सेइंग? डिफरेंट कलर हमने दिए हैं तो डिफरेंट डिफरेंट ये कंडीशंस हो सकती है, हमारे वेरिएबल्स हो सकते हैं, ये उनके लिए होता है। और अब हम उनकी ग्रुपिंग कर रहे हैं कि कम एक्सप्रेशन है या ज़्यादा है। माइक्रो एरे में वैसे किया जाता है, लेकिन हियर वी हैव टेकन द डिफरेंट वेरिएबल्स, तो उसका जो डेटा निकल के आया व्हाट इज द एसोसिएशन, उसके बेसिस पे हम ग्रुपिंग कर रहे हैं, रीग्रुपिंग कर रहे हैं, मतलब क्लस्टरिंग करना। तो अब हमारे ये निकल के आए, हियर यू कैन रीड दिस ऑल्सो, यहाँ पे दिया है इसमें एक्सप्लेनेशन, इसको मैंने पढ़ा नहीं है बट it is written here actually in the G what they are doing, results can, हम रेप्लिकेशन करते हैं, एक्सटर्नल रेप्लिकेशन, इंडिपेंडेंट कोर मीन्स इंडिपेंडेंट है, वो जो कोहॉर्ट में आया था ना निकल के, उससे इंडिपेंडेंट एनालिसिस है। ये कोहॉर्ट में निकल के आए हमारे अलग अलग वो बन गए, फिर से हम एनालिसिस कर रहे हैं, पोस्ट जीनोम एनालिसिस कर रहे हैं, बाद में एनालिसिस कर रहे हैं ना, पोस्ट मतलब GWAS एनालिसिस, ये हमने कर लिया, उसके बाद में क्या निकल के आया उसको हम एनालिसिस कर रहे हैं। ठीक है, तो इसका जब हम लेके चले थे स्टार्टिंग में, उससे कोई रिलेशन नहीं होता है, हम देखते हैं कि क्या निकल के आया है, व्हाट हैज कम आउट बाय द एनालिसिस। इसको आप ऐसे समझ सकते हो, यू यूज द डिफरेंट ग्रुप्स, यू डिफरेंट बुक्स स्टडी आप करते हो, सेम ग्रुप आप सेम, I am giving you the same lecture and I am giving you the same notes, but the different student will get the different marks, they will perform differentially। ठीक है, तो मैं एनालिसिस कर सकती हूँ, हमने सेम क्राइटेरिया पे दिया था ना, आई एम एनालाइजिंग यू ऑन द सेम प्लेटफॉर्म, बट वेरी कर सकता है, सम पीपल, सम स्टूडेंट में स्टडी डिफरेंट बुक्स, वो अपने आप से स्टडी कर रहे हैं, सेल्फ स्टडी कर रहे हैं, तो उनकी परफॉर्मेंस क्यों इंक्रीज हुई, उसकी अलग अलग वो हो सकते हैं। ठीक है, सो इट डिपेंड्स। तो एनालिसिस में निकल के आ सकता है कुछ और आ सकता है निकल के। तो वी नीड टू फाइंड आउट द कॉजेस, और ये हो सकते हैं। सो SNP's वेरिएशन हम यहाँ फाइंड आउट कर रहे हैं। सो इट डिपेंड्स कि हम इनपुट क्या दे रहे हैं, वो तो हमारा होता ही है, उसके बेसिस पे भी हमारे डिफरेंट ग्रुप निकल के आता है ना। जब हम एग्ज़ाम लेते हैं उसके बाद में स्टूडेंट्स अपीयर इन द एग्जामिनेशन, दे गेट द डिफरेंट मार्क्स, दे हैव द डिफरेंट परफॉर्मेंस। तो ये वो जो एग्जामिनेशन रिजल्ट है, मार्कशीट, उसमें परफॉर्मेंस जो आपने एग्ज़ाम में दी है उसके बेसिस पे ग्रुपिंग होती है, फर्स्ट डिवीज़न, सेकेंड डिवीज़न, थर्ड डिवीज़न, दैट इज़ दैट ग्रुपिंग। ठीक है, लेकिन वो भी हम क्लासिफिकेशन एक कह सकते हैं कि हमने ग्रुपिंग कर रखी है पहले से ना, कि 60% से ऊपर आएगा तो उनको फर्स्ट डिवीज़न में रखेंगे, इस तरह से है, तो उसको भी हम क्लासिफिकेशन ही कहेंगे। लेकिन जब हम कोई और क्राइटेरिया यूज़ करें, डिफरेंट न्यू क्राइटेरिया, उससे एनालिसिस कर रहे हैं, दैट इज़ कॉल्ड क्लस्टरिंग, और इस तरह के एनालिसिस होते हैं। वी आर यूजिंग सम न्यू मेथड्स और क्लस्टरिंग, कुछ डिफरेंट क्राइटेरिया यूज़ करके on the basis of their performance। ठीक है, we are trying to cluster them together। तो ये है आपका एनालिसिस जो बाद में हम फाइनली करते हैं। Have you all understood? Now lecture is over. If you have any query you can ask। Okay, because at the starting we don't know, because why we are doing this study, to know something, to reveal the causes। तो ये हम इतना सारा एनालिसिस करके हमें पता चलता है कि क्या डिफरेंस होता है। ठीक है. Initially we classify the different organism on the basis of phenotypic appearance कि मंकी जो है ह्यूमन के जैसा दिखता है वो एक क्लास का हो गया. लेकिन जब हमने when we obtain the sequence, when we have done the comparison, then we come to know कि माउस भी बहुत सिमिलर है ह्यूमन के, जीन बहुत सारे सिमिलर हैं. तो उस तरह से हम ग्रुपिंग करने लगे ना. तो इसे आप कह सकते हो कि पहले नहीं पता था ना, इनिशियली वी डोंट हैव द सीक्वेंस. ठीक है. तो जब हमने डिफरेंट क्राइटेरिया यूज़ किया तो वो हम क्लस्टरिंग में कंसीडर करते हैं कि प्री डिफाइंड नहीं होता है. क्लासिफिकेशन प्री डिफाइंड होता है, क्लस्टरिंग प्री डिफाइंड नहीं होती. इट इज़ बेस्ड ऑन द रिजल्ट्स व्हाट वी विल ऑब्टेन, उसके बेसिस पे भी होगी. तो कई बार डिफरेंट, जैसे अफ्रीकन, अमेरिकन, हम ये एज्यूम कर सकते हैं कि वो भी एक साथ में ग्रुप हो जाएं किसी बेसिस पर. ठीक है. सो दैट विल बी रिवील्ड आफ्टर द एनालिसिस. आर यू गेटिंग व्हाट आई एम सेइंग? आर यू गेटिंग व्हाट आई एम सेइंग? ओके. लेक्चर बहुत बड़ा हो गया है. It's too lengthy. Okay. Now lecture is over. Okay. Already I took more time. Okay. You can ask the questions. You can type in the chat box also. Hello, there was disconnection. Okay, I have recorded a lecture. So have you understood everything? Okay, I will provide you the recording of this lecture. So if you have any question, you can ask. I have explained you this figure. We were discussing about that. Okay. So finally, by all the analysis, we come to know what are the different groupings can be done after the analysis. Whatever revealed in the analysis, on the basis of that. That may vary what we have taken at the starting. So different populations can show the relation with each other. Like the African can show the relation with another population. So now these are the different populations, but they can show the relation. So, okay. Do you have any question? Do you have any question? Okay, you can type your attendance in the chat box. Wait for computational biology and bioinformatics. Okay. So just write down your attendance in the chat box. Okay. Okay. So stopping the recording. I'm going to stop.
Excuse me, ma'am.
Yes.
Ma'am, I have some queries regarding the practicals.
Yes.
So मैम आपने जो मतलब टॉपिक्स दिए थे तो वो टॉपिक्स एक बार कंफर्म कर दीजिए मैम, बहुत कंफ्यूजन है.
नहीं, I have provided you the list of the practicals. So if you are not caring able to do the difficult one, there was a small easy exercise that we have done at the starting. So there are the three different types of the practicals. So you can choose the easy one. if you're not able
Okay,
to,
ma'am. So all
or you
we
can
have to
ask,
write is the.
or you can ask if you have more interest, then you can contact me, because now
Okay,
your
ma'am.
syllabus theory is completed, but if you want to do more or some practical or some project work, we can do it also.
Okay.
Okay.
Okay, ma'am. Sure. So all we have to write is aim and steps. That's it.
Yes or no, objective, hypothesis,
Okay, ma'am.
methodology, result,
Okay, ma'am.
observation,
Okay, ma'am.
then conclusion. Okay.
Okay, ma'am.
Okay, I will send you the format. Okay. Although I have
Okay,
told
ma'am.
you in the class, I have already explained in the class, but I will,
Yes,
if
ma'am.
you
But we
want,
have a
I
few
will send you.
confusions regarding the
I will post you the
formats.
format. Okay. You just write
Okay,
down
ma'am.
in that format and send it to me.
Okay, ma'am. We'll do the same.
But
Thank you, ma'am.
make the practical file. I think okay, genome wide. ये GWAS method now it is clear to everybody, and I have an lectures also for the association, so you can attend that also.
Okay, I'm sure.
Okay.
Thank you, ma'am.
Okay, so now we will do means the only the practicals. Okay. In the next class we will do some practicals, then you will, if you have any difficulty, but just now you just submit your file whatever you have done. Okay, you just submit it to, you just try to means learn something, then I will tell you how you should improve. Okay.
Okay,
Now,
ma'am. Ma'am,
now,
all the practical work would be done in practical notebook or practical file.
but just write down in the MS file, then I will you just submit it, I will tell you what are the errors and what improvement you can do in that,
Okay, ma'am.
and then you just means do it on the hard copy or in the
Okay, ma'am.
practical file. Okay.
Okay, ma'am.
But first
Thank
that's why
you.
I'm asking to submit you the MS Word file in the Google Classroom.
Okay, ma'am. Got it.
Okay. Then or you can write down your doubts or question in the Google Classroom. Okay. Whatever means,
Okay,
do
ma'am.
not write me on the WhatsApp group. Okay, just do not mail me, just submit it in the Google Classroom. Okay, in your class.
Sure, ma'am.
Okay, because every all the record will be there. We will keep the that record. Okay, so in the email, I mean, it is very difficult to search for all the student or in the WhatsApp. Okay, so lecture is now over. Means everybody has done the attendance. So already I took the lot of time of you. Just type your attendance in the chat box if anybody is left. Now, okay. Okay, class is now over.